Okay, so our next speaker, Katherine Chau, and she'll be talking about land use and social environment on bee development. Thank you.
Thank you. Hi, everyone. My name is Katherine. I'm a postdoc in Sandra Ray Hen's lab here at York University. Today, I'll be talking about two projects that I recently published where we explored a wild bee's microbiome in the context of environmental change and environmental stress. So I think by now we're well aware that there's many environmental factors contributing to global bee population declines, some of which are like agriculture, urbanization, and spread of bee diseases. But not only do these environmental factors obviously directly negatively impact the bee, they're also impacting the various microorganisms that reside on or inside the bee. This is also known as the microbiome. And this can also pose some serious problems when we're concerned with bee health. So as a broad definition, the microbiome consists of all the microorganisms that reside in a particular environment. When we're studying organisms, the organism itself is the environment we're concerned with. And in bee microbiome research, most studies will focus on bacteria, fungi, and viruses, as extensive research has shown that many microbes and strains within these three domains can be quite detrimental to bees if left unchecked. But there's also many microbes, especially in bacteria, involved with important biological processes like digestion, nutritional uptake, immunity, even cognitive ability. And dysbiosis, which is the imbalance of a healthy microbiome, often results in poor development or increased mortality. And so observing microbiomes has become quite important in bee conservation research. So how do we explore them? So most studies will target loci that will specifically just amplify bacteria, viruses, or fungi. But metagenomics is an approach that allows us to identify all other non-target species DNA that happen to get sequenced at the same time as the target species. So in both of my projects, the whole bee specimen was used for either whole genome or whole transcriptomic sequencing. We obtained sequence reads, which are then mapped against a reference genome. Anything that is unmapped, we can extract and then classify them for taxonomy. And this lets us identify simultaneously all the other organisms that are interacting with our target bee or species. And what's great about this approach is that it also lets us explore organisms beyond just the three main domains, bacteria, fungi, and viruses. So in my projects, I study Ceratina calcarata, the small carpenter bee. Ceratina is a globally distributed genus, and Ceratina calcarata is a native wild bee found here in North America. This bee likes to burrow in dead woody stems, plants such as raspberry or sumac. It's classified as a subsocial bee in that nest consists primarily of a mother and her offspring. The offspring will develop while the mother remains, and she'll perform other important duties like cleaning and guarding. So unlike social bee microbiomes, which is heavily shaped by the interactions between colony members, the microbiomes of subsocial and solitary bees are heavily derived from and influenced by their environment. And so they make great model bees to explore how wild bee microbiomes may be responding to their environment. So this brings me to my first project, where we use the metagenomic approach to explore Ceratina calcarata's microbiome alongside its population structure for individuals that were sampled along an urban gradient here across the Greater Toronto area. So my co-author, Evan, had gone out and sampled 180 females by trekking through sites that had a lot of desirable nesting habitats, so a lot of woody stem plants. The females were captured and they were used for whole genome sequencing. Any of the sequence reads that were mapped were used for popped-in analysis, which was done by our other co-author, Farida. And then anything that was unmapped, I used for microbiome analysis. And this is a map just showing the sample sites across the GTA as well as the urban gradient. So the darker the circle, the more urbanized or developed that site location is. And here we were expecting evidence of subpopulations to exist in our bee. One, because with increasing urbanization, natural habitats become more fragmented. And so this limits gene flow. But two, our bee is a very small, cute bee, and small bees have very limited dispersal flight ability. And so it becomes more difficult for them to traverse across very fragmented urban landscapes. And we also expected their microbiome composition to change along the urban gradient because the differences in environment across these different sites should result in different presences and abundances of the various microorganisms that coexist. And our results were interesting. So first and foremost, PopGen analysis found that despite urbanization and being a small cute bee, Serotina calcarata in the GTA was actually found to be one large population. So to better understand this, we decided to map dispersal corridors most likely to be used by Serotina calcarata. So these would be corridors or pathways rich in vegetation land features, so crops, trees, shrublands. And when we map these vegetation-rich corridors, which are all the red squiggly lines on the map, you can see that in the center area where there's a big blur, those corridors are actually very limited. And this makes sense because in this area, urbanization is quite high. But we can still see that there are some corridors between the circles, the blurred circles, which are our sample sites. And so this suggests to us that Serotina calcarata in this landscape is very likely using these highly limited vegetation-rich corridors to traverse across the urban landscape, and likely why we did not see subpopulations. When we look at the microbiome data, we did not actually see any significant shifts in the composition across the urban landscape, but we did observe that taxa richness often increased in sites that were very characteristic of dense urban areas. So these would be sites that had, of course, high developmental rates and low open green space, but also high local temperature and low precipitation levels. And we noticed that it was more pronounced for bacteria. So as bacterial richness increased, as urbanization increased. And this can be a problem because it means that exposure to potential bacterial pathogens is also increased in these sites. So when we then explore the average number of well-known potential bee and plant pathogens in our data set, we found a clear cluster of higher pathogen density in those sites that have those high urban or dense urbanization characteristics. And so with this project, we learned that vegetation-rich corridors, even when they're limited in urban landscapes, are quite important for urban bee dispersal, especially for small bees. And we also found that urbanization in this sample region increased microbial richness, especially for bacteria. And this poses a risk because it also means there's a higher pathogen load with increasing urbanization. And this project is great because it really highlights the conservation power of integrating these different methods. So integrating metagenomics with PopGen, we saw that while we did not observe our urbanization to impact Serotina calcarata's population structure, we did see that it had some meaningful impacts on its microbiome instead. And so moving on to my second project, we again applied the metagenomic approach, but we do it for transcriptomic data. In this project, we were interested to see how gene expression and microbiome composition change in individuals that were raised with or without mothers, so care and no care. The lack of mothers here represents the environmental stress, since they're no longer keeping the nest clean and sanitary. We were also interested to see how this changed across development. So here's an image of some of the developmental phases for Serotina calcarata. There's 19 distinct phases, but we grouped them into 4 major ones: early, late, pupae, and callow. And then my co-author Miriam had collected over 100 sumac and raspberry stem nests from the field. Half of the nests were opened up, eggs were taken out and reared to different stages in the lab. This represents the no care group because mothers are absent. The other half, individuals at the different developmental stages, were immediately taken out, and they represent the care group because they were already raised naturally in the wild with their mothers. They were used for RNA extraction to analyze gene expression with the help of our other co-author, Jesse. And again, any unmapped reads were used for microbiome analysis. And so this is just a volcano plots, and there's a lot of info here, but I just want to sort of summarize it quickly. So these show how many genes were significantly upregulated, which are denoted by the red and blue dots. And just in general, what we see is that although we see evidence of significant upregulation in all stages and care groups, we see that maternal care had the strongest effect on gene expression during early larval development. There was many more upregulated genes at this time, and as well as it was the highest in the no care group. So in this group, this was the only category that had significant enrichment for transcription activity. And that itself is actually an important defensive mechanism because it helps to regulate homeostasis as well as prioritize developmental processes. when we look at the microbiome data, we see a similar pattern. So there was there was greater amounts of microbial detections in the no care group during early larval development. So the no care are the black bars. And then when we separate the taxa into their distinct groups, we see that fungi and bacteria drove most of the variation. And then when we take a closer look at these two domains, in both groups, taxa were more abundant in no care early larvae, again, the black bars. And then finally, the most abundant taxon we found that significantly contributed to microbiome variation between no care and care early larvae was the plant pathogen Aspergillus. It's known to cause stonebird disease in honeybees and plant rot in a lot of plant species. It was overabundant in no care early larvae. And while its effects are not quite known exactly for Serotina calcarata, our findings showed how easily imbalanced the microbiome of stressed individuals can become. And just in conclusion, so we found in this project that maternal deprivation affected no care early larvae the most due to upregulation of genes involved with gene regulation itself, as well at the same time, microbiome data showed that environmentally stressed individuals are more susceptible to bacterial and fungal outbreaks during the early larval stage. And in this project, it shows how, again, integrating these various methods can highlight important dual responses of stressed organisms. So in this case, both methods highlight that in Serotina calcarata, early larvae are easily the most stressed, get easily more stressed are easily more environmentally stressed out. And with that, I'd just like to thank my supervisor, Sandra Rahan, the Rahan Lab, and my co-authors for help with this project, as well as all the funding partners and York University for, again, also helping with this project, making the research possible. And if there's any questions, I'd be happy to take them. Thank you.